Good afternoon, everyone, and welcome to the NINDS Biomarkers Program webinar. My name is Ashley Gibbons, and I am the Program Operations Specialist for the Biomarkers Program. I'm going to introduce our panelists for today. We have our program directors, Dr. Mary Ann Pellimonter, who is our program lead. We have Dr. Carol Taylor-Birds, who is our FOA's point of contact. And we have our health program specialist, Dr. Genevieve Lind. How today's webinar is going to run. We are going to present our slides, and then we'll have a question and answering period once the presentation is over. This webinar is recorded, and we will send out slides and recording after the webinar is over. So a few days after the webinar, you'll get an email from me with the slides and the recording. Also, if you have a question that's related specifically to your application, please email us your question along with your AIMS page, and we will set up a time so that you can meet with someone on our team. Thank you guys so much, and I'm going to turn the webinar over to Dr. Carol Taylor-Birds at this time.
All right, thank you, Ashley, and thank you all for joining us today. So just to start with a little bit of background, the NINDS Biomarker Program was launched in 2017 in order to accelerate the process from biomarker discovery to biomarker validation in a manner consistent with FDA evidentiary criteria guidelines. So this starts with initial discovery phase, where you'd identify the biomarker, develop the detection method using standardized samples, and pilot an initial clinical validation study. Before moving on to definitive analytical validation and clinical validation. So FDA qualification is not a requirement of the program, but it is designed to develop the package of evidence that would be needed if you were to submit. And the end goal really is to have validated biomarkers for use in clinical trials or in clinical practice. So we currently have 3 sets of funding opportunities that are designed to identify and validate fit-for-purpose biomarkers. And starting with PAR 19315 for the initial discovery activities. So again, including identification of the biomarker or development of the detection method with an initial proof of concept clinical study. And these might be using samples from humans or data, retrospective data, or an initial clinical prospective study. And then these are followed by PAR 21056 and 057 for analytical validation activities to optimize and rigorously evaluate the performance characteristics of the biomarker's detection method. And then finally, PAR 21058 and 059, which are for conducting full-scale multi-site clinical validation studies that are needed to establish the degree to which the biomarker identifies or predicts the concept of interest within a defined clinical context of use. So note that the next submission date for all of these is June 22nd, which is quite soon. And then after that, the following receipt date will be February 22nd of 2022. So I'm going to go over all of these in a little bit more detail. But before I do, we want to go through a couple of things that are really important for all of these FOAs, starting with the definition of a biomarker. Right, so a biomarker is defined as a measurable indicator of normal biological processes, disease progression, or response to a therapeutic intervention. So in order to help clarify and standardize the terminology for biomarkers, the NIH and the FDA developed the Biomarkers, Endpoints, and Other Tools, or BEST glossary, in 2016. And that's used to classify biomarkers as pharmacodynamic response, diagnostic, prognostic, predictive, susceptibility risk, monitoring, or safety biomarkers. Surrogate endpoints would also be in this, but there's substantially more evidence required for validating a surrogate endpoint. So the BEST glossary provides brief descriptions of each of these biomarker categories, and it's important to familiarize yourself with these definitions before applying to any of the FOAs here, because the expectations for what evidence is needed really depends both on the category of the biomarker and the context of use. So we're going to spend a minute on the context of use, because again, this is a critical concept. And context of use is a concise description that includes both the category of biomarker and how it will be used in therapeutic development or in clinical practice. Just to spend a minute to go through a few examples here, you might want, if you're developing your biomarker, you might have a clinical context of use where you're proposing to develop a prognostic, diagnostic, or predictive biomarker to stratify or enrich patient populations, right? And your study design is going to be slightly different depending on which of those biomarker categories it is. Or you could be developing a pharmacodynamic response biomarker for demonstrating therapeutic target engagement and informing dosing decisions. Or maybe you're validating a susceptibility risk biomarker for indicating the probability of developing a particular disease or condition. So as the name suggests, in general, monitoring biomarkers are used to monitor changes in a condition or changes in response to a therapeutic intervention or environmental exposure. And nearly all of the biomarker categories could potentially be used to inform treatment decisions, but of course, there's going to be additional, the expectations and safety and risk-benefit assessments will be different for a clinical practice than something going to be used in a clinical trial. So any of these categories and context of use are within the scope of the biomarker FOAs that we're talking about here. And of course, you're going to ask, you might have a candidate biomarker that could be used in several different contexts of use, but you want to be careful about specifying which context of use you're validating your biomarker for, because your study design should really match what your proposed use is, right? So we describe this program as product-driven, because the validated biomarker signature is the product, and you're trying to develop the evidence to validate your product so that you know how it can be used, what it tells you, and what degree of certainty you can trust what it tells you for that use. Now, there's a lot to do within a confined budget and time period, and so it tends not to be feasible to propose more than one or two contexts of use at a time. So it's essential that you really decide what's the most important, justifiable, and feasible context of use to aim for. So moving on to the PARs themselves. This is just a list of the currently available funding opportunities that I'll be talking about in more detail. But just to highlight a few differences in the mechanism. So the Discovery PAR is an R61, R33. And then we have both a academic U01 biomarker for analytical validation and for clinical validation, and then an associated small business fast track U44 option as well for both the analytical and clinical validation. So to talk about the mechanism, our PAR 19315 is a phase mechanism. And so that means that the transition to the R33 phase is dependent on achieving go, no-go milestones. And note that the total grant duration cannot be more than 5 years, but there's flexibility in how many years are dedicated to each phase. And it's a good idea to include a timeline for the activities proposed in both phases, right? Because you want to demonstrate the feasibility of achieving the project goals within that time period. Technically, there are no hard caps for budget, but any application that's requesting more than 500,000 direct costs in any 1 year must contact us at least 6 weeks before submitting the application, because we need internal review at NINDS for approval by leadership before these can come in. So you need permission to come in with budgets over that size. Now, to talk about the mechanism just a little bit more, the go, no-go criteria are really critical between the R61 and the R33 phase, right? Because you need to have extremely clear, quantitative, and definitive milestones that say, this is what has to be achieved, and this is the level of performance you need to see or accuracy in order to justify moving on to the R33 phase. And that process will be a stop point where we'll internally review the progress and decide whether or not it can move on or not. Now, for the analytical and clinical validations, PARs, as I said, all of these are either U01 or U44 options. The project can be up to 5 years again, although some of the projects really only need 3 or 4 years, especially in the analytical validation stage. And just like the discovery PAR, any applications to the academic U01 FOAs having budgets that over the 500,000 direct costs per year have to have prior approval to come in. So again, reach out to us at least 6 weeks before the receipt date. So the U44 option is for SBIR fast track applications, and you can request up to 700,000 total costs per year in phase 1 and up to 1.5 million per year in phase 2. But of course, those budgets need to be clearly justified. And again, for all of these cooperative agreements, you need milestones at the end of each year. And so the annual review is fairly stringent between moving from one year to the next. And again, these milestones need to be extremely clear, quantitative, and definitive. And milestones to include, and these are just some examples, you could have more than that, and you have whatever is appropriate for the study, but you're gonna need to include some performance milestones, including things like the target sensitivity, specificity, and accuracy, and precision threshold you need to reach, and then what the acceptable dynamic range is for the context of use you're proposing. And in some cases, signal to noise might be appropriate. Demonstrating standardization across sites, maximal acceptable intra and inter coefficient of variation, specification of data quality assurances, and quality control standards. So these are just a few examples that you should think about it carefully about what's necessary for your project. And then also progress milestones, such as enrollment or sample and data collection goals, finalization of protocols, SOPs, contracts, and consultation with the FDA if that's the route you're going. So when we say quantitative with clear go, no go criteria, just to do a really simple example, assay reproducibility will be tested is not a good milestone. Say an assay will be repeated 3 times at each site by independent users with intra and inter coefficient of variation less than or equal to 50% will be considered acceptable. Okay, that tells us very clearly what it is you need to meet and how you're going to meet it. So let's talk about the FOAs themselves, and I'm going to show sort of a comparison of all 3 for different parts of these PARs, just to sort of compare how they differ. So looking at the outcome goal, because you always want to think about the outcome goal for these PARs, what do you need to have by the end of that 5 years? So for discovery, you have to have completed the initial clinical validation and detection method, development of a biomarker or biomarker signature, and refine the context of use. So for discovery, you might come in with a couple of contexts of use that you're thinking about, and you want to figure out how good your biomarker is between 1 or 2 different contexts of use, or something that's a little bit more refined at the end. And the entry criteria for this include biological rationale, supporting the concept of a candidate biomarker or signature, the relevance for the neurotherapeutic development or clinical practice, demonstration of knowledge and technical proficiency for the detection method. All of these things are fairly standard. And of course, this needs to be within the mission of NINDS. And NIA is also signed on to this PAR, but not the others. So within the mission of either of those is appropriate for discovery. For analytical validation, again, this is to have, by the end of that period of performance, your performance characteristics of the detection method, and establish the technical protocols or process pipelines for reliably measuring the biomarker or biomarker signature for a specified clinical context of use. So for the entry criteria, of course, it includes everything within the discovery, plus you want to have the defined context of use, and you want to have evidence of preliminary clinical validation of the biomarker. You just want to justify that this biomarker is ready, that there's evidence that it is associated with a clinical concept of interest, and that there's good reason to do the analytical validation. And then evidence that the detection method has at least undergone initial development, right? So this is not a device development or a assay development, FOA. You can do optimization, but you really need to have a working prototype that you're going to be optimizing. And then finally, for clinical validation, here is really where you're establishing the biomarker and biomarker signature acceptably identifies, measures, or predicts the concept of interest within a defined clinical context of use. So again, really important to define your context of use. And also it includes all of the entry criteria listed above. Plus you also want to have evidence that the analytical validation of the detection method has already been completed, right? So you might be coming in for a clinical validation using commercially available analytical validation detection method that's already been analytically validated. And that could be appropriate, but you need to justify or explain how it's been validated and is appropriate for your clinical context. So looking at the application characteristics for discovery, again, you're going to propose a context of use or uses, and you want to have a strong justification for the biomarker and the detection method. You want to address feasibility, including the potential added clinical burden and cost of the methodology that you're using, the modality. And the outcomes used to validate the biomarker and biomarker signature need to be clearly stated and justified. Again, these need to be well defined. You need to have the go, no go criteria between the R61 and R33. And animal studies are allowed in the R61 phase, but they really need to be carefully justified. And reviewers will comment if they feel like it's not efficiently getting to the validation that's needed, right? So those can get cut, or you might be dinged for proposing something that isn't necessary. And then human samples and data are encouraged in the R61, but they're required in the R33 phase. Now, for analytical and clinical validation, animal studies are not appropriate for either of those mechanisms. And for analytical validation, multi-site study design is expected. Now, there are exceptions, so there might be a scientific justification, like in an extremely rare disease. But those need to be, if you're not proposing multi-site, you need to justify why. And again, these have annual milestones and need to be conducted in using human samples, data, retrospective data, or prospective study designs. And then again, with both this and the clinical validation, these have defined context of use and similar application characteristics in clinical validation. So what kind of metrics would you be thinking about for evaluating your biomarker, right? So in discovery, this is going to overlap basically with both analytical and clinical, since there are activities within discovery that are the early stage phase of both of these. So accuracy and precision, analytical sensitivity, reportable range of results, harmonization of the analytical performance and method of detection, and so on, right? And so analytical validation, these, you're also looking at reference intervals or controls and calibrators, establishment of appropriate quality control, and improvement procedures that are needed. And then clinical validation, right? You really need to demonstrate the clinical sensitivity and specificity of the biomarker within the context of use. And you might have binary or continuous analysis as appropriate for your particular biomarker and measurement. And then area under the curve as determined by receiver operating characteristics analysis. And really that should be done in all of these as well. But this is going to be in the final clinical validation of it. And then establishing the appropriate cutoff or thresholds for the biomarker in order to make decisions using it within that context of use are going to be essential. And then estimation of the prevalence of the marker within the subjects or patients that you're intending to use this biomarker with. And so, of course, positive predictive value and negative predictive value would follow. So what's not responsive to these PARs? So that would include anything that's a development of a therapeutic agent or devices as the primary intent. So it's not for therapy development. We have other FOAs at NINDS that are targeted for that. And then evaluation of clinical efficacy, effectiveness, clinical management as the primary intent is also not appropriate. Natural history studies that don't include the biomarker identification and validation would not be appropriate. Of course, studies that fail to include milestones and studies outside of the mission of NINDS will be administratively withdrawn, right? So we can take these out prior to review. So just to sort of reiterate the points we've talked about for each of these funding opportunities, right? When you're developing your proposal, think about it for discovery. You know, are you proposing something that by the end of the 5 years, will you have a candidate biomarker or endpoint that has a developed detection method and a preliminary validation in human samples or data by the end of the grant period? And for analytical validation, will you have completed definitive analytical validation, including establishing the performance parameters of the detection method? Will you have developed the technical protocol, analytical pipeline, depending on your biomarker, for reliably measuring the biomarker signature for the intended context of use? And in clinical validation, will you have demonstrated the clinical sensitivity and specificity and positive predictive value and negative predictive value for the intended populations for the proposed context of use, right? So you really want to think about what is the intended population and do you have appropriate representation? And then finally, will the biomarker signature be ready to be used for the intended research or patient care setting? So just before we go to Q&As, I want to make you aware that we have a biomarker website at NINDS, and there's additional information here, including the projects that have already been funded, which you can see in this blue box on the right that are through NIH Reporter. There's a number of programs and resources at the bottom of this page that are not shown that are good to be aware of, things like sources of samples from BioSend, list of the funding opportunities, and then two things just to point out. We have some frequently asked questions that will probably continue to update as more questions come in, but that's a good resource to sort of check as you're thinking about your projects. And then there's also a table of just sort of a quick reference guide for some of the other funding opportunities at NINDS that do allow biomarker validation studies, but might be slightly different in their entry criteria approach. So it's a good tool to sort of just check where you want to go. And with that, we are happy to answer some questions.
All right, thank you, Carol. I'll be directing questions to Dr. Taylor-Birds and Dr. Pelley-Mounter. We'll get started with the first question. Carol, for the R31, R61, R33, where can people find information for the grant document preparation guidelines, such as the page limitation, font size and style, line spacing, etc.?
Right, so thanks, Genevieve. So the information and links are in the FOA. So if you go into PAR 19315 and go down to the information on how to submit the application, you'll see a table that tells you about the mechanism and the page limits for those. And you can also go to NIH's grant website, and there's additional information there too. But if you have a specific question, you can always just email us.
Great, thanks, Carol. Marianne, does the PIR 19315 require a commercialization plan to be developed?
No, it doesn't actually, because PAR 19-315 is not specifically set up for SBI or STTR grants. So because of that, it doesn't require it. But businesses obviously can still apply to that particular PAR.
Great, thank you. Carol, by our definition, is clinical validation including animal pharmacology models or exclusively human clinical?
Yeah, that's exclusively clinical for this. That would be, that could be appropriate for some of the other programs at NIH, but not for these PARs.
Great, thank you, Carol. And just as a reminder, if you have additional questions, please put them in the Q&A box. Thank you. Marianne, can you explain the difference between a biomarker and a biomarker signature?
Yeah, so when actually a biomarker could be a biomarker signature. So you're talking about subsets there, sort of. So when we talk about a biomarker signature, we talk about multiple biomarkers that, when put together, provide a more accurate picture of what the physiology is reflecting in terms of clinical output. So often a biomarker signature is composed of perhaps several omics markers or perhaps a multimodal marker. And usually then you have to think about how the physician will use that, a complex biomarker like that. So very often machine learning and algorithms are used to develop a one type readout that actually is the signature.
Okay, thank you. Carol, for the R61, R33 team building, is it preferred to have the co-Is involved in both phases? In other words, should a specific expert whose role is primarily helpful in the R61 also be justified for the R33, even if that person's expertise is not directly related to that phase of the study?
So the expertise should really match what the plans for the proposal are. So it would be possible to only include specific expertise in the R33 phase, but it really just depends on what you're doing in each of those.
Right. Marianne, could you please explain in more detail the harmonization of analytical performance of the method of detection process for the discovery phase?
Yeah, so that refers to the idea that when you're using any type of method of detection, you have to keep in mind the end goal, which is going to be eventually use of that detection method in any place that, makes sense in terms of how you're going to use it. So for that reason, operators, machines, all of that have to be as standardized as possible so that there's no room for error just based on using a different machine or different detection method. So that's one of the reasons Carol mentioned that even in the analytical validation FOAs, it is really important to do multi-site type validation if it's called for in view of the type of detection method. But you want things to give you a reliable readout. So that's the idea behind that.
Great, thanks, Marianne. Carol, for the R61, R33 transition, do applicants need to separate out budget projections just by period or also separate by R61 from R33 costs?
So this is a regular research project grant, so it's going to be annual budgets that you're going to be describing and justifying that way. Your aims are going to be distinct, so you're going to propose aims for R61 and aims for R33, but the budgets are going to be annual.
All right, Marianne, for these funding opportunity announcements, what percentage of the work can be outsourced?
There's a specific percentage related to what the actual business will do. If it's an SBIR, as you recall, 2 of the validation FOAs are SBIRs, so they have their own instructions that are outlined in the FOA. But if it's one of the U01s or the R61, R33, there is no specific percentage.
All right, Carol, there's a requirement in the FOA for a letter from tech transfer from all participating institutions for the R61, R33. What is the extent to what is needed in this tech transfer letter, especially in the situation in which there's no planned development of new intellectual property?
Right, so The tech transfer letter, really you need to just describe whether or not it's necessary. I think I just lost the question. Can you repeat that, Genevieve? Sorry.
What is specifically required? What is the extent of information that is needed in that tech transfer letter, especially if there's not any new intellectual property that is going to be developed?
Yeah, you really just want to describe what the landscape is. I'm not sure I'm fully answering the question. I don't know. Mary Ann, do you have a better answer?
Well, I'd just say there's one additional thing to address, and that is whether or not you have permission to use things like, let's say you're developing an ELISA assay of some sort and you're using specific antibodies. Do you have permission to use those antibodies? Are they in the public domain? That kind of thing. Same way if you happen to be using genetically modified animals in the very beginning of your R61, just make sure that any reagents, items, ideas that you're using, you have permission to use. And often it's a good idea to, in addition to, providing evidence that you have permission, also, like have letters from the people who might, if they actually, people who own, let's say, a reagent. So it's just to make sure that you are free and have permission to use what the reagents you're using.
Thank you, Mary Ann. So Carol, for a company that qualifies for the Omnibus SBIR opportunities, are the chances of success using the PAR 19.315 about the same as the U44 SBIR fast track mechanism?
I wouldn't say there's a difference in chance, right? You're competing against other applications. You're trying to get a competitive score that's justifiable for funding. The budgets are different, so the funding for SBIR is distinct from the funding for the regular R61, R33, or U01s. And so there can be differences in availability of funds, but there's no hard and fast rule about which is more or less competitive.
All right, it doesn't look like we have any additional questions at this time. If you have more questions, please put them in the Q&A. We'll hang out for a couple of minutes if anybody comes up with any additional questions. Looks like we got another question, Mary Ann. What is the SBIR total budget for this?
It's actually listed in the FOA. And honestly, I'm sorry, I can't remember exactly what those numbers are, but you would see it on the cover sheet of the FOA.
Yeah, so it's up to 5 years of funding, and you can ask for up to 700 total costs for the phase 1 and 1.5 for the phase 2. And if anyone would like to unmute themselves and ask a question, or would like to ask a question live rather than typing it in, please let us know. Yes, this is being recorded and will be sent out.
All right, looks like we have another question. Do we have any suggestions on how to identify an advisor, or coach, or an expert with biomarker expertise to help a team through the biomarker development process? Marianne, I'll hand that question to you.
Yeah, so we don't have a formal method to do that. We don't have like formal consultants or advisors, but I think probably the best thing to do, as Carol has mentioned, is just email us and we'll set up a call with you. And usually it won't be just one of us. We'll have the team on the call. And if, for instance, I know you're involved in a business, we could have one of the SBIR people there to provide any business related advice, as well as we can provide the biomarker development process advice.
So if you do have additional follow-up questions, do please reach out. Happy to talk to you and set up a call. I'll talk to you about draft games, as Marianne said. And if you have any follow-up questions after the webinar, please feel free to shoot us an email.
You're very welcome, actually, by the way. We enjoy giving these webinars, and we really do want you to reach out to us. We really want to help you because we, really want to have some decent biomarkers that can be used by clinicians.
Well, thank you guys so much again for attending our webinar and asking questions. We will also send you a copy of the slides. And once we have the recording of the webinar, we'll send that out as well. Thanks again and have a wonderful afternoon.